Good morning, everyone, and welcome to today's MPG Primer. Today we have a great talk on structural variation in human genomes by a lab that is at the forefront of algorithms for this, the Telkowski lab. Today we have Xuefang Zhao and Emma Pierce-Hoffman, and I'd like to just give you a brief introduction to their background. So Xuefang is an instructor in the Telkowski lab. She has contributed to the development of GATK SV, the cloud-based ensemble method to identify structural variation, or SV. She has led the Genomic SV discovery effort in the recent coverage 1000 Genomes project samples, the recent Human Genome Diversity Project, and is now leading the SV discovery in Nomad version 4. Xuefang holds a master's degree in statistics and a PhD in computational biology from the University of Michigan at Ann Arbor. Emma is a senior computational associate in the Telkowski lab as well, and is in the methods team in the data sciences platform. She contributes to method development for the GATK SV pipeline for structural variant calling. And from short read whole genome sequencing data. She also leads the short read whole genome sequencing SV call set generation for the All of Us research program. And she received her BS in statistics and data science from Yale University. Because this is such a great topic and has so much information, we're going to have a lot of content today. So we're going to ask to have questions just at the end. All right. Thank you guys so much.
Thank you. Hey, can people hear?
You just need to use this one.
Oh, okay. Hey, everyone. My name is this good? Okay, my name is Xuefang. Today, I'm really excited to share with you a brief concept and our work in structural variation in human genetics. I'm going to first very briefly introduce genomic structural variation, which I'm pretty sure most of you are already familiar with, so I'll go through very fast. And then I'm going to generally talk about the genomic SV discovery saying in this field in the past like 1 to 2 decades. And then I'm going to focus on our recent study in Nomad before SVs. And I'll also briefly talk about the relatively newer long-read sequencing and how that technology has brought into our field for SV discovery. And I'll then hand over to Emma to tell you this really exciting algorithm named GATK SV, which we used to do all those SV discovery in our study. So as we all know, there's SNV, single nucleotide variation in human genomes that has been very well studied. There has been many very awesome methods represented by our GATK. Well, except for SNV's genomic variation is actually more diverse. A lot of them are much bigger. So there's what we call indels that represent small deletions and insertions that are usually we consider them as the events between 2 and 50 bases. And beyond that size, there's a whole class of structural variations, including the copy number variations, such as like a deletion of a piece of our genome, a duplication of a copy, or like multi-allelic CNVs where there are sites in the genome that you can see the copy number ranging from like 0 to very crazy numbers like 100 or so. Except for that, there are also insertions that includes novel insertion, which means like insertion of novel sequences, or there are also other insertions that are of known categories, most are mobile elements, including the ALU, L1, SVA. Those are the subtypes of mobile elements that are still active in human genome. And there are also inversions, complex events, and interchromosomal translocations that our algorithms are now able to identify. So I hope to briefly generalize the massive investment of this field into SV discovery in the past like 1.5 decades. Back in 2010, when the Southern Genome Pilots project just published their first paper, they sequenced 179 whole genome sequences. They generated SNV, indel, and SV calls from them. And at that time, they were only able to identify about 20,000 SV sites across as many samples, across as many genomes. Well, during this past like 14, 15 years, there has been significantly increase in the amount of investment into this field. Like we're generating more and more whole genome sequences. We're making better and better algorithms, and such that we were able to identify more SV sites with better performance from the genome, and which allows us to study the general rules of SVs and their distribution across human genome. So here I listed a couple of studies that kind of, for me, is like the landmark of this field, where for 1,000 genomes, they increase from 100 to 1,000 to a couple thousand. Then when it comes to Nomad and all of us, our mass capacity are at almost 10,000 samples in the All of Us phase 2 study, and a little bit over 63,000 samples in Nomad V4. By the way, the Nomad V4 study were actually, the SV COSA were generated and cross-genotyped across over 140,000 genomes. But because some of the samples are related, some of them lack proper consent from the donors to release their data. So we finalized the releasable data set to include about half of the samples. So the number here. And in terms of the SV loci that we are now aware of that exist in human genomes, they increased from 20,000 to now in Nomad V4 I have about 1.2 million SV loci. At the same time, the quality or the power of our SV discovery method keep increasing as well. So you can see from the very early times, the algorithms found like 3,000, 4,000 SVs per genome. While now, with better methods, and I hope to highlight GATK SV, where Emma is going to tell you every details about, but the Nomad, the HSVC, not HSVC1, sorry, 1,000 genomes high coverage, the Nomad V2 were all generated with our GATK SV ensemble method. And we can, meantime, we can identify more and more diverse types of SVs. So now I'm gonna try to focus on the recent Nomad V4 SV COSA that our lab has greatly contributed to. This data set, the releasable set, is relatively diverse. You can see that only less than half of the samples are from non-Finnish European, while the rest half, they distribute across all those populations, most of which are previously, underrepresented in our other studies, including the African, the Asian, the Jewish, Middle Eastern. And from those samples, we identified about 1.2 million SV loci. Vast majority, or half of those SV loci, are deletions, which they distribute very frequently in the genome, and also that is the variant type that is relatively easy for short reads to identify, which I'll show you later. Except for that, we also have good amount of duplications, insertions, and other types like inversions and complex events. And this is how, in general, the structural variation, the size of structural variation distributes. In general, the size of structural variation is reversely correlated with their frequency. So bigger SVs are less frequent, of course, because bigger SVs are more likely to cause deleterious consequences. They may require more energy to form, so they're less observed. But I want to highlight the three still active movement types in human genome, human genomes include ALU, L1, and SVA, where you could see the purple color here represents insertions. So we usually observe a big number of ALUs around 300 bases, SVAs around 1 kb, and the L1 around 6 kb. Those are peaks in the size distribution, but overall, this is what we were expecting. And if we zoom into the individual genomes in this Nomad call set, we define an average of a little bit less than 12,000 SVs per genome. And the colors is consistent with the previous set. The red here represents deletions, which is mostly frequently discovered, followed by duplications and insertions. And when we compare the number of SVs carried by each genome by like different population, it is consistent with the SNV index studies that African populations carry the most amount of SVs than the rest of the populations. But the question is, so as I showed before, the GATK SV is calling a couple times more SVs than other data set, right? But what is the quality of this algorithm? Like, how do you trust all those new, like, novel sites that are found by this algorithm are real? So we did a very serious QC on them. The first, here I'm showing you the direct comparison of the Nomad V4 dataset versus a couple others, relatively recent population scale studies, including Nomad V2, Southern Genomes, CCDG, and the recent TopMed study. So I understand this figure might be a little bit overwhelming to look at, but let me explain. So the left side of each panel shows you the Nomad V4 SVs, and the color bars shows you the proportion of Nomad V4 SVs that is overlapped by the other study by different comparison criteria. So the blue is like in the other data set, you have an SV that directly overlaps with it. And if the pink represents you don't have a direct overlap, but you have something very close that they might be the same events, but you just, for some reason, couldn't call them very clear. And I'll explain the other pages to say like why, like sometimes we cannot call them very accurately. And on this right side of the panel is vice versa. So that is the proportion of SVs in this other data set that is overlapped by the Nomad V4 data set. So you can see in general, like we covered most of the SV loci that are already detected by another study, meanwhile we also have a lot of new novel loci in this study that were not covered by others. So the next question is, what is the quality or what is the precision? Like well, it is good news that we call a lot more variants, but what if they're all wrong, right? So good news is there are more and more long-read sequencing data available. And in this data set, we included Southern Genome samples, and we do end up having 59 related samples that were sequenced by the Human Genome Structural Variation Consortium that have the long-read PacBio Hi-Fi data available. So we benchmarked our data set, our SVs, with the long reads by two different methods. The first is the algorithm named VPER. This is the algorithm I wrote during PhD training. So it just took the long read, directly compare the structural variation, tells you whether it's real or not. The other method we use is we overlap our SVs by the SV calls generated from the long read data that were published in Ebert et al. In Science. So and SVs that are overlap that are supported by either method would be considered real. So overall, the precision were pretty nice that over 90% of deletion insertions were supported by PacBio. By PacBio. And when it comes to duplications, inversions, those are trickier types of SVs to find for short reads. We still have like 70 to 80% precision. Well, this is not the full story. So I showed in like a previous paper that short read Illumina sequencing or short-read parent sequencing, they're great at calling SVs in most of the genome. But when it comes to the highly repetitive segmental duplicates and the simple repeat regions, they're terrible. Like they couldn't find a lot of them, and we couldn't have very good point precision. So we split those calls by their genomic context to say if they're in the repetitive regions or not. We find that when we restrict to the relatively clean genomic regions that consist over 90% of the genome, our precision was pretty high, 95% or higher. So this is very reassuring to let us know that our quality were very decent, low false positive rate in the data. And this is a very brief look into potential functional impact of SVs on human genome. So we So like about 140 genes per genome were interrupted by loss of function. And this is very comparable to our Noman V2 data. Well, as I just showed, our best capacity of SV calling are at 12,000 SVs per genome from short reads. But when it comes to long reads, the first three bars on the top, they can easily find more than double of the SV count than we do. So the next question would, of course, be like, what are we missing? Like, how do we use the long read to guide better SV discovery from short reads? There has been a couple long read activities since in the recent years. So the first paper the GSVC published were back in 19, where they deep sequenced 3 trails with multiple technologies, and then they later enlarged it to 35 genomes. And they generate the cost. So we have matched short reads data for all those samples. So we can do like sample to apple comparison in the same sample. So what we find is that first, here, I want to show you, even in the same genome, short read and long read find total different things. Like only 1/3 to 2/3 of their SVs overlaps. But why is that, right? That's the question. So here I'm showing you, I think the key is at the repetitive sequences, where you can see the pink and the yellow colors are the highly repetitive genomic sequences. They consist 99.7% of the genome. And when it goes to the coding regions, especially the coding region of the important genes, they're even less, they're like 2 to 5%. However, SVs are enriched in those regions. On the right, I'm showing you how the short read SVs distribute across those regions and how the long read SVs distribute. So after we split the variants by the genomic context, we could see that in the relatively clean genomic region, see how high the overlap is. So deletions in the clean genomic regions, they share very good they share very good overlap with the long read costs. But when it comes to the repetitive regions, that's where the long read really shows superior sensitivity at. And meanwhile, like we're a little bit limited in the discovery of insertions and duplications. So a couple take-home messages from this study that I hope to highlight is that our current short read data can detect deletions in over 90% of the genomes very well. That's very comparable to long read data. When it comes to the repetitive genomic regions, that is where short read lack power, while long reads are really good at. And for insertions, short reads also lacks some capability, but I think there's a lot of rooms for further method development to make this better. Oh, and one more thing I hope to highlight is our study from this. So our finding from this study is that there is one, black hole in the current long read algorithms are the long read, so the large CMVs, like CMVs when it comes to over 10 KB and above, you could see long read cannot really find them. Well, this is not a technical limitation, but rather a algorithm limitation, because long read usually use assembly method, which it'll be computational heavy to assemble through such a long region to find those events. But I think there are also a lot of room for method development, and our GATK CMV method are already shown to be pretty good at make this cost. So to summarize, I think the long read field is for a promise that there are more and more long read sequences available from the HESVC, the HPRC, and also this very famous SAS1463 family. They're now extending it to this is a Utah family that they have 4 generation. So you have many trails, you have many siblings to compare against. They recently do high coverage, PacBio, high precision, high fidelity sequences on all of them, and Oxford Nanopore I think the first 3 generations. So a lot of data to use here. And also our colleagues at Southern Genome Project recently published their, actually last Thursday, they published their preprint over 1,000 ONT data on Southern genome samples, and all of us phase 1 has published over 1,000 lower coverage high-fi genomes. I'm going to skip this summary and hand over to Emma to tell you everything about GATK SV. Oh, and this is my acknowledgement. Thank you.
All right, thank you. So now that Xuefang has introduced what are structural variants and what are some of the developments in the field over the past decade or so, I'm going to focus much more closely on our methods that are available for detecting structural variants from short-read whole genome sequencing data, specifically focusing on our GATK SV pipeline. So first, let's talk about how structural variants actually show up in short-read sequencing data. So if you have reads that are just about 150 bases long, you can imagine that a structural variant, which could be hundreds, thousands, or millions of bases long, is not going to fit in that single short read. So instead of just looking at the sequence of the reads, we use 4 classes of evidence to identify SVs in short read data. Those are split read, paired end, read depth, and B allele frequency. And I'm going to go through each of those in detail. We'll start with split read or SR evidence. So I have a diagram to kind of explain what this is. The sample 1 on the top does not have a structural variant in this region. And you can see that the reads align well to the reference from that sample. But sample 2 on the bottom has a deletion of the middle piece of this region compared to the reference. And the reads only align partially back to the reference. So we're illustrating that by the red portion of the read represents the mismatch between that read and the reference. That's also called a soft clip or a split read. And those occur because that deleted region does not exist in sample 2. So those reads span from the left to the right side of the deletion. But when we attempt to align them back to the reference, we now have that deleted region present. So that there is a mismatch between what we expect to see in that location compared to what we observe in the sample, and we get split reads. And an important thing about split reads is that they can actually tell us the precise location of the structural variant breakpoints. So you can see from the diagram, the dotted blue lines show the exact start and end of this deletion because we can just identify where the soft clips start, where those mismatches start. So in that way, split reads are a powerful type of evidence for SVs. So let's look at a real life example. This is a heterozygous deletion that was identified in a real person. The deleted region is shown by the blue bar at the top. And on the right side, we've zoomed in on the 5' end, and you can see that there are soft clips shown by the rainbow of mismatches at the end of some of those reads. And those are split reads. So we can see in IGB that this deletion has SR evidence. Next, let's talk about paired end or PE evidence. So with Illumina sequencing, we sequence from both ends of a sequencing fragment. Those ends are called mates, and they form a read pair. And we expect the paired alignments to be oriented inwards. And the insert size or the distance between the mates is expected to be close to some typical size, close to the mean. So when we observe deviations in the insert size or the orientation of those reads, that can indicate the presence of a structural variant. For example, this deletion has a larger insert size than the normal or no event example. And this tandem duplication has the reads oriented outwards instead of inwards. So let's take a closer look at an example of a deletion to kind of understand better why that's happening. So again, sample 1 does not have a variant at this location, and the read pairs have a typical insert size for this sample, but sample 2 does have a deletion compared to the reference, And the alignments are much farther apart than expected. And that's because sample 2 does not contain the deleted sequence. So when you go and sequence reads in sample 2, those fragments are the size that you would expect. But then when you align them back to the reference, the deleted region is there, and that pushes them farther apart. And one thing to note about PE evidence is that it gives us a lot of information about different types of structural variants. We can really learn a lot about what kind of rearrangement has occurred, but it doesn't tell us the precise location of the breakpoints. You can see, looking at the diagram, that you can get a pretty good idea of where this SV is, but not the exact start and end. So again, let's go back to our real life example. Here in IGV, we have colored and sorted by insert size. So all of these read pairs in red have a larger than expected insert size. And you can see that they're placed much farther apart than the read pairs that are in gray. So those are showing the PE evidence for this deletion. Our next type of evidence is read depth, or RD. So changes in read depth or coverage can signal the presence of large copy number variants. So going through these examples, in the case of a heterozygous deletion, we see a drop in read depth over the deleted region. And that's because there's only one copy of that DNA in the sample as opposed to two. So there's just less DNA to sequence. In the case of a homozygous deletion, there's a complete lack of coverage over that deleted region. It doesn't exist, we can't sequence it. In the case of a heterozygous duplication, we see an increase in coverage or read depth across the duplicated region. So read depth can be very helpful for discovering large CNVs, as Xuefang mentioned, that especially those that we currently are less able to discover in long reads. And again, we can take a look back at our real life example. The coverage track at the top of this IGV screenshot shows the read depth profile across the sample, and you can see that it does dip to about half over the deleted region. So this deletion does have read depth support. Our final type of evidence is B allele frequency, or BAF. This is also just known as allele fraction. And this refers to the fraction of reads that support a SNP. And in the case of a heterozygous single nucleotide variant, you would expect that to be around 0.5. But in the presence of copy number variants, that allele fraction can shift. So in our heterozygous deletion example, when we have SNPs on the non-deleted haplotype, the allele fraction for those SNPs is 1, because there's actually only one copy of that region, and so all of the reads should support that SNP. In the case of a heterozygous duplication, when we have SNPs on the haplotype that is not carrying the duplication, then we observe an allele fraction of around 1/3. So 1/3 of those reads carry that SNP, and the other two copies have the duplication. So you can see that B allele frequency can signal or support the presence of copy number variants, but that signal is a little bit fuzzier than some of the other evidence types. So in GATK SV, we only use BAF for variant filtering rather than variant discovery. So let's put it all together in our real life example in IGV. You can see the SNPs represented in the coverage track at the top. And for all of the SNPs in the deleted region, you can see that the allele fraction is 1. So in fact, this heterozygous deletion has all 4 types of evidence. Okay, so now that we know what to look for, let's talk about how we use those pieces to identify structural variants. Our GATK SV pipeline is a computational pipeline for germline structural variant discovery from short read whole genome sequencing data. The inputs are Illumina deep short read WGS, and as an output, we get a genotyped and annotated SV VCF, along with ploidy estimates and supporting QC reports. So I'm going to go over some key elements of our methods. First, our ensemble approach, variant merging, joint genotyping, complex event resolution, and functional annotation. So I'll start with a schematic of the bird's eye view of those pieces, and then we'll go through them each in detail. So GATK SV takes an ensemble approach, employing 5 different SV discovery algorithms and utilizing all 4 of those evidence types we discussed. We then merge variants, joint genotype all sites and all samples. We perform complex event resolution, functional annotation, resulting in a call set with high sensitivity and high specificity, as Xuefang showed. And this pipeline is designed to run in the Google Cloud platform, and in particular in Terra. So first, coming back to evidence classes, we saw that each type of evidence has different strengths. So we want to leverage all 4 of those evidence types for the best sensitivity and specificity, and to discover SVs across the whole range of sizes and types. But there isn't a single SV discovery algorithm that leverages all 4 evidence types at once. So GATK SV runs multiple SV calling tools. And in that way, we take an ensemble approach for optimal sensitivity and specificity. So let's take a look at what are the SV discovery tools that we use. First, Manta is a really good general purpose SV algorithm. We use MELT, which specializes in mobile element insertions, and WAM has high sensitivity to small duplications that tend to be missed by other algorithms. So using multiple tools increases our sensitivity. Note that none of these that I'm showing right now actually use read depth, so we have to add in 2 other colors for that, which I'll get to next. Also note that MELT has a restrictive license, so we're working on switching over to an open source alternative called Scramble. Okay, so I mentioned that the previous tools don't take advantage of read depth, so these are the CNV colors that we run. First is GATK germline CNV color, or GCNV, and then CNMOPs. And both of these are depth-based CNV colors that require a batch of samples. They do joint calling. And using both of these methods together improves our sensitivity and segmentation. So you can imagine that now we've run multiple SV discovery algorithms across many samples, and we may have discovered the same structural variant by multiple algorithms and in multiple samples. So we want to now reduce our redundancy. But SVs are not always determined, especially from short reads, with single base precision. So we might have highly overlapping variants that are not exactly the same. They don't share the same breakpoints, but we still want to merge them because they likely represent the same underlying structural variant. So what we do is we cluster those highly overlapping calls shown in black, and we collapse them into a single representative variant shown in red. Then we can move on to joint genotyping and assign genotypes at every site to every sample based on the PE, SR, and RD evidence. And similar to in SNP and indel calling, this increases both our sensitivity and specificity. We can rescue calls that may have been called in one sample but missed in another, and we can learn the evidence distributions across samples. The next step is complex variant resolution. So a complex structural variant is one that has more than one SV signature. An example would be a duplication and an inversion together. And there are many different ways that complex SVs appear in the human genome, but GATK SV specifically resolves 11 subtypes of complex SVs, which are shown here. Now that we have all of our structural variants, we want to understand their functional impact. So we perform functional annotation. We're trying to answer the questions, what genes and non-coding elements does each variant overlap? And what impact do we predict the variant would have on protein function or transcriptional regulation? So let's go through a quick example. Here, the alternate allele carries a single exon deletion in this gene compared to the reference. And our SV annotation tool would indicate that there's a probable loss of function in this gene because a large portion of the coding sequence has been deleted. That's likely to have some kind of impact on the function. Okay, so here's a quick recap of the methods that we just went over. GATK SV takes an ensemble approach to improve sensitivity and specificity. We merge variants to reduce redundancy. We perform joint genotyping of all sites across all samples. We resolve complex events, and we perform functional annotation. I also want to note that there are two ways that you can run GATK SV. The first is cohort mode, in which you apply GATK SV to a group of samples to produce a joint call set. And the second is single sample mode. So maybe you don't have over 100 samples on which you want to perform SV calling, but you still want to be able to run GATK SV on one sample at a time. And you can do that. And the way that we do that is we use pre-computed reference panel data to be able to take advantage of the improvements that we get by considering multiple samples at some of those joint steps. And importantly, GATK SV single sample mode is currently available publicly as a Terra featured workspace to make it really easy to run. We're working on getting a featured workspace available for cohort mode as well soon. So single sample mode is recommended when you have fewer than 100 samples, so not enough to run joint mode. So that is also ideal maybe in a clinical setting where you might have rolling sample sets. You don't have all of your samples at once. Single sample mode is faster and easier to run, but with the caveats that it's more expensive per sample and there is lower sensitivity. It also requires reference panel resources that were generated with cohort mode, and we provide a publicly available pre-computed reference panel using data from the 1000 Genomes project. And with that, I will acknowledge the whole Takaski Lab and Broad SV team, particularly those listed on the slide who contributed to methods development, as well as the consortia who provided data and funding. And I think we have time for questions. And I think questions for both talks.
While we wait for the people in the audience to come up to the mics, we have a few questions online. Someone is asking anonymously, can I use GATK SV on HPC?
Yeah, so currently GATK SV, we only support Google Cloud as a backend. We do have some users who have been attempting to get it set up on an HPC. It really does take advantage of the parallelization that's available in the cloud. So currently we don't support HPC as a backend, but you're welcome to try.
There's also another question by Satoshi. Does GATK SV accept BGI samples?
I don't know the answer to that.
I think as long as they're like short read, pair and short read whole genome sequencing data, GATK SV could process them. And in general, the same as every other algorithm, the cleaner the data is, the better quality you will get from your data. So I think BGI data could work as long as they're pair and short read whole genome sequencing.
Presentations. I'm curious about the best way for detecting SVs. Like long read sequencing is great for most SVs, but misses some that are detected by short reads. and short reads detect some things that long reads don't? Do we need a combination of short read and long read to get comprehensive SV detection, or is one method better? What do you think we're going to be doing for SV detection in the next 3 to 5 years?
That's a really great question. Thank you. So in my view, that's my personal view, I think long read are very informative. We could use them as a nice guidance to further improve our power with current short reads, because think about how many short reads we already have available, 100,000 from all of us, over 100,000 from Nomad. We can use all those data through short reads. Well, long reads is like we can use them as a guidance, but if for a project you have enough budget, of course you're welcome to deep sequence both long read and short read. That'll give you best performance. So a couple other activities that we're considering. First is for long read data, you can genotype them into short read data. So for the sites that were not detected by short read but only detected by long read, you have the short read data. You can find those sites and genotype them to say if the short read sample carry them, but those only works for common variants, right? So the more long read data we have, the more rare sites we can get into, but still you cannot get as many as rare as with short read data. The other consideration is to do a combination of high coverage short reads, maybe low coverage long reads. So the idea is to use the long reads as a backbone. So you can align your short reads, maybe assemble them in a way to further improve the capacity.
Great, that's very helpful. Thanks very much.
Thanks for the great talk. So quick question, I guess, about the repetitive regions and difficulties in detecting things there. So I guess I was wondering how much of this is alignment issues and shortcomings of like HG38 as a reference that, I don't know, has there been any interest in trying to use T to T or things like this? So I guess just how much is like fundamental statistical limits versus how much is just the tools currently aren't able to capture these repetitive regions?
That's another great question. Thank you. So that's what we've been thinking about all the time. But I think, of course, alignment difficulty is the issue because think about short reads, they're usually one read is 150 bases, while there are a lot of repetitive sequences that are way bigger than that. So when you have such a read and you have so many different locations in the genome you can align to, how do you know where you go to, right? That is the advantage of long read, where they're long enough so they can find the anchors in the unique regions to align that read to. In terms of T2T, there are a lot of new studies using T2T, including the short read studies, and they do show improvements in SV qualities, but we haven't officially transferred to T2T because the concern is there may not have been enough annotations in the genome. So a lot of time you need to do leftover. The question is how accurate is that leftover? I cannot say, but I feel like my personal opinion is if there's a better reference and if there's good enough annotations with that reference, we should transfer over to say if we have better performance. But I think the repetitive region would always be an issue with the short read data. Oh, one more option I forgot to mention for the previous question is, I think one more option is to target the repetitive sequences with long read, which only takes 10% of genome. But how easy is that? That's a technical question because they're 10% of genome, but they scatter everywhere around the genome. So how many probes you need to design? We don't know. But it's something very interesting to explore in the future.
Makes sense. Thanks so much.
Can I ask one more question? Today we've been talking a lot about germline SV calling. Can you guys give us a broad outlook on what this looks like in somatic? I know we'll have a somatic talk next, but what's your take on somatic SV calling?
Yeah, that's a great question too. That's something we've been considering. Somatic is hard. I think our lab used to have a presentation a month ago that Chris presented. He has a method to define somatic variants from NIPS, the mother's blood, that the pregnancy woman's blood, where they were able to define low mosaic level variants. that's really hard, and we're really limited in our capacity. But I think with better data, with better technology, there might be a day in the future that we can detect all those somatic variants. We can find all those stream crypsis events in the cancer genomes in the near future. Maybe not near, but
Let's thank our speakers one more time.